Hi, everyone. My name is Derek. I'm a business development manager here at Genobiotexas. Thank you for everyone for coming today to today's webinar. We're excited to have you for today's talk titled A Hypoxia-Responsive tRNA-Derived Small RNA Confers Kidney Protection via RNA Autophagy. Before we get started, just want to very quickly introduce Genobiotexas for those who might not be familiar. So at Genobiotexas, we're committed to advancing biomedical research by providing innovative and high quality animal models and services that can help accelerate the development of next generation therapeutics. So first, before I introduce the speaker, at the end, we're gonna have a question and answer session. So if you have any questions, feel free to drop them in the chat or ask them at the end. So I'm thrilled to introduce our speaker today, Dr. Gopeng Lee. Dr. Lee is an assistant professor at the Massachusetts General Hospital and Harvard Medical School. After graduating from Tongji University in Shanghai, China, he completed his postdoc at the Massachusetts General Hospital in 2021. He's the recipient of the American Heart Association Postdoctoral Fellowship Award and Career Development Award. He has also won the Young Investigator Awards from multiple organizations. His current research focuses on the regulation and roles of non-coding RNAs, particularly transfer RNA-derived small RNAs in cardiac, metabolic, and neurological diseases. And he's funded by the American Heart Association and the NIH. Without further ado, let's get started. Dr. Lee, the floor is yours.
Thank you. Thank you for the kind introduction and the invitation for sure. So it's my honor to present our first functional tDR study in the kidney. So first, this is my disclosure. I'll give some background of the tDR first. As you know, the transfer RNA we cut as tRNA-derived small RNAs is derived from the transfer RNA. When transfer RNA has been identified as mRNA decoder for more than 50 years, and there's about 500 tRNA genes in the human genome, with 50% of them are actively transcribing. So these results in that the transfer RNA is the most abundant RNA species in the cells. In 1990, some investigator identified in E. coli that these tRNA can be cleaved into smaller fragments. But until 2008 and 2009, that several independent groups identified that these cleaved fragments are not simply tRNA debris, but they have distinct functions. The first one reported is about the tRNA fragments we call tDRs could regulate the translation in different behaviors. So it's a little bit challenging, I would say, because I mean, this tRNA research has been postponed until 2009 is because of the modification on the tRNA molecules. As you know, every tRNA molecule has around 13 different chemical modifications, and especially those methylation will present a lot of challenges for tDR research. It starts from sequencing to detection and gain of function, loss of function, even, at least the most challenging part of therapeutic applications. Let's start from the sequencing part, because as you may know that when you sequencing using the normal small RNA-seq to identify previously microRNAs, you can rarely capture the tRNA molecules. No one has questioned why, but the truth is most of the tRNA has a huge modification on these, especially the methylation on different positions, especially this M1A at position 58. These methylation will stop the reverse transcriptase when you do normal RNA-seq. So you will never capture the tRNA reads or you can rarely capture a very small part of a tRNA reads in your sequencing data. Until 2015, Tolu, our collaborator from UCSC, developed a methodology using pretreatment of RFKB, which is a demethylase, to remove the methylation, then facilitate the reverse transcription go through the methylation positions, which facilitate the detection of tRNAs. Until that, more and more advanced tools, sequencing techniques developed to identify much more tDR rates. So as you can see, it's called DMTRNA reads, which using mutated alpha KB could demethylate these other two M1G and at different positions. And even now, the autophagic from UC Berkeley has using a power switch reverse transcriptase that can jump over these methylation stops to facilitate the detection or sequencing of full-length tRNA reads. So there's tons of tDR identified through sequencing with these advanced sequencing techniques. And they have been implicated in different biological processes, including transcription, translation, and stress granule formation, and some epigenetic regulation as well. But most of them are focused on in the tumor cancer biology, and some in the immunology, very rarely in neurological disease and cardiometabolic diseases. So that's why when my fellows postdoc training started, located in the cardiovascular research center. So we are interested in how these tDR are regulated and what's the function in ischemia heart disease, which is the leading cause of death worldwide since 2000. So in our first tDR profiling paper, we have used multiple in vitro cell culture system, including several genome, the HEK cells, human embryonic kidney cells, vivo cell type, which is cancer cell type. And in addition to these cardiomyocyte and cardiofibroblasts, we treat them with 3 ischemia reperfusion related stresses like nutritional deprivation, hypoxia, and oxidative stress. And that project, we profile both the cellular tDRs and extracellular tDRs upon different stresses. It's a huge data set. I will not go through detail, but if you're interested in just check this paper published in Advanced Science in 2022. So today I want to focus on because when analyzing those stress-regulated tDRs across different cell types, it results thousands and thousands of tDRs, both in cells and extracellular vesicles, in different cell types under different stresses. So the first one we want to know to study the functions of them. The first one we focus is HEK cells inside the cellular tDRs. So when we check in the tDR profiles between different stressors in HEK cells, we identify the hypoxia-induced the most abundant tDR change. So as very interestingly, when we check the top regulated tDRs, especially when the HEK cells, the hypoxia treatment groups, the top 2 tDRs are all derived from the same tRNA. It's called tRNA SVGTC, with one from the 5' end and another one from the 3' end. So we first validated using northern blot. And to further confirm that this is the fragmentation, which means the cleave from the tRNA for tRNA instead of transcription itself. So you design this self-quenched tDR biogenesis reporter by conjugating the fluorescence group and quencher group to either end of the full-length tRNA. So this reporter can only release fluorescence signal upon cleavage. As you can see here, hypoxia induce a lot of significantly increased FAM signaling in the cells, indicating that these generation of these fragments are cleavage based. So next we want to know what's the function of them. We synthesized the mimics of these top two tDRs and transfect them into the cells, checking all the 3 major hypoxia-related stress response, including ER stress, mitophagy, and autophagy. Neither of them has a regulation of ER stress and the mitophagy, but the 3' tDR has a very abundant regulation on the autophagy pathway. As you can see, the increased lipidation of LC3B and downregulation of P62. So as you may heard, that when studying autophagy, it's a very complicated process, which includes it's an autophagy flux, which includes several steps, including the formation of autophagisome, the fusion with lysosome, and the maturation then eventually goes to the degradation. So to rigorously study what's the regulation of this tDR on autophagy flux, we use multiple approaches suggested by the autophagy study guideline. The first one is the autophagy flux reporter, which has a tandem RP-GP fluorescence protein conjugate with LC3B. So all the autophagisome will be labeled by both RP and GP, but when the autophagisome fuse with lysosome, the GP signaling will be silenced. Only the RP signaling will sustain. So with this cell type reporter, we have seen when we transfect the TDR mimics to these reporter cell lines, as you can see here, the tDR mimics, full-prime tDR, induce both the increase of both RP-GP double positive and RP single positive cells, punctate-like structure inside the cells. This is the statistic analysis. So this is autophagisome, and this is autolysosomes, indicating this is increased autophagy flux. In addition to this, we have used beflomycin to block the autophagy pathway. So when you block the flux, nothing getting degraded. So the accumulation will indicate the overall activity or levels of autophagy flux. So what we can saw here is with the beflomycin treatment, the lipidated LC3B is even more than the control groups, indicating that these tDR, 3' tDR, enhance autophagy flux. So we have the gain function. The question is, people usually, the review is challenging, like how you demonstrate the functionality you proved using tDR mimics is true inside the cells. So we are trying to do some loss of function studies to block the functionality of the endogenous tDRs. This is the RNA antisense. We call it ASOs. Antisense oligonucleotide to block the expression and the function of this full-prime tDR. It has been widely used since 2015 by several groups, demonstrate the feasibility of knocking it down using ASOs. This is the RNA antisense ASO we designed it to. We have some algorithm to predict the best ASO can target the tDR itself without disturbing the full-length tRNAs. And this is knockdown efficiency we proved. It doesn't have a significant effect on the full-length tRNAs, but significantly downregulates, knock down the full-prime tDRs. What we saw here is when we transfect these ASO into the HEC cells and expose them into hypoxia condition, you still see an increased level of lipidated LC3B. As I mentioned to you, the autophagy is not a simple process that can solely determined by the lipidated LC3B. This probably due to a blockage of autophagy flux as well. To further confirm this, we use two different strategies similarly at the gain of function. With this autophagy flux reporter, as you can see here with the hypoxia treatment, there's a significant increased amount of RP single positive punctate-like structure in the cells, indicating an increased autophagy flux. But when we transfect the ASO targeting these 3' tDR, most of the signal are RP and GLP double positive, means these are autophagisomes, and which fail to maturate to autolysosomes. As you can see here, there's a significant decrease of the autolysosome numbers. Further, when we use beflomycin to block the autophagy flux and checking the LC3B lipidation, as you can see here, it has a decreased amount of LC3B lipidation, indicating that when you're knocking down the 3' TDR, it blocks autophagy flux. So next we want to answer, because these are all done in HEK cells, and HEK cells has some similarity to kidney cells. So we want to know what's the function of this in vivo. Before doing anything, we expanded our analysis using a mouse TDR tissue atlas from our collaborators group. And we found that this 3' TDR is highly enriched in kidney, heart, and brain. As you may know, that kidney is functional hypoxic. So we first check the expression of this 3' TDR among different human primary kidney cells. That's quite a lot. I would say among all of these main or key kidney cells, this TDR is abundantly expressed across all of these, especially in the epithelial cells and kidney fibroblast. So next we want to know what's the function, because they already have a very high abundant baseline level of this 3' TDR. So we're directly knocking them down using ASOs. As you can see here, when we knock it down, it seems block has a very significant effect of the autophagy flux. It blocks the autophagy flux, as you can see, by the decreased amount of LC3B lipidation. In the glomerular epithelial cells as well, the same phenotype, it blocked the autophagy flux. When we culture the cells for a longer time, the cells seem to undergoing apoptosis pathway, as seen by the clipped Caspase-3, and increased cell death and decreased cell viability. How about in the kidney itself? So we have using different mouse models to check the dynamic regulation of this 3' TDR in kidney tissues across different stresses, like the volume overload, which included a unionectomy model. As you can see, after 1 hour of the surgery, this 3' TDR goes up. And with the proteinuria, we did the high concentrated BSA injection for a week. This 3' TDR also goes up. Surgically, we have done obstructive nephropathy, which is unilateral ureteral obstruction. As you can see, 4 hours after surgery, this 3' TDR goes up and is sustained for a higher expression over 10 days. So next, we want to know what the function of these 3' TDR in vivo. With the SOs, it has an advantage with some modification, especially the EPS backbone modification. It will facilitate the uptake by the cell itself. So all of the ESOs we can directly inject a sub-Q or retroorbital or tail vein. They can be absorbed by the tissues. And by doing this continuously injection, we are able to knocking down this 3' TDR in the kidney tissues. This is the ASO inject groups in the UO model. As you can see in the kidney, is significantly knocking down the expression of this 3' TDR. Similarly, when we check the RNA, because we check in the kidney injuries, as you can see here, even in the shame group, when we block this 3' TDR, the kidney injury markers like KIM1 and GAL is increased. It's even worse when we do in the UO groups with the knocking down of the 3' TDR, it worsened the kidney injuries. As shown by the kidney injury markers, the inflammation and fibrosis. The histology data also confirm this. As you can see, there's higher apoptotic cells in the kidney and have higher immune cell infiltration and fibrosis in the kidney tissue as well. We also check the Western blot to confirm the similar phenotypes. In addition to these apoptosis, inflammation, and fibrosis, we also check the autophagy pathway. It's consistent with our in vitro studies. The autophagy flux is blocked by the ASO inhibition, ASO administration, which block the 3' TDRs. So we were being asked by the reviewer saying, no, you have to show some gain of function studies. It's very challenging to deliver TDR mimics or deliver any RNAs into the kidney tissues. So we have been luckily tested several nanoparticles, which is two actually, lipid-based and polymer-based commercial ones. Very, very interestingly, we found that only polymer nanoparticles can deliver somehow the tDR mimics into kidney. We have been able to optimize it using different dose, different formulations, and be able to deliver a significant amount of tDR into the kidney tissues. So then for the UO model we previously used, it's a irreversible mouse kidney injury model. It's very challenging to reverse the fibrosis in these kidney tissues. Instead, we're choosing a moderate kidney injury, which is a renal ischemia perfusion injury. We only ligate the vein for the kidney for around 20 minutes to induce some ischemia and perfusion injury in this kidney. And most likely this kidney will be recovered after a week. So this is the first characterization of this expression of this 3' tDR within these IR injury models. As you can see here, immediately or very earlier phase after the ischemia, you will observe an increase of this 3' tDR. But afterwards, when the kidney function reversed back and all the stress is gone, this 3' tDR expression has also went down. So in these models, we delivered on administered the tDR mimics every other day through intravenous injection. This is the overexpression efficiency. So then we check the kidney functions, first some qPCR like chlorophyll, which is a reno-protective factor or secretion proteins. This one is significantly higher in the tDR administered kidney tissues. Very significantly, as you can see here, the kidney injury markers like KIM1 and NGAL is significantly downregulated in these tDR administered groups. Same with other inflammation markers and fibrosis markers. Histology also showed the consistent results like the apoptosis, kidney cell injury, and fibrosis. Same, we did the autophagy pathway examination using Western blot. As you can see here, the autophagy flux, I would say the autophagy flux also increased by the administration of these 3' tDR mimics. So confirming these tDR has a very strong reno-protective roles in the kidney. When you block it, it worsens the kidney injuries. But when you deliver it or increasing its abundancy, it protects the kidney from injuries. So the next question we want to ask is, how does this 3' tDR activate autophagy flux? The first we want to know is to identify the key structural motif in these tDRs. Very likely when we presented in the Harvard RNA Institute seminars, there's one group from Brigham identified that your tDR has a very guanine-rich motif. Previously, his group, together with our CAO, identified previously two tDRs that has terminal oligoguanine motif form this very specific structure called intramolecular G quadruplex motifs, which is 4 strands to form this G quadruplex motifs. And when we check the sequence of our own tDRs, it has very guanine rich, especially with 2 4-guanine motifs here. So we collaborate with them and try to identify whether these 3' tDR can form G4 structures. This is a very simple experiment, the gel shifting assay, that you assemble these 3' tDR within this G4 preferred environment with potassium or lithium, which is non-G4 perform allowance conditions. By comparing those two, you will identify these bands, extra bands, which is around 120 nucleotide, which is just 4 times more than the tDR mimics, which is 33 nucleotides. So it's indicating that form a G quadruplex within 4 different intro tDR strands. This is the control identified by their group with tRNA, ALA, AGC 5' tDR, this one. Similarly, when we try to identify whether these 4 guanine motifs are required for the formation of this G4 structure, we mutated individually or simultaneously. As you can see here, when you mutate individually, it kind of lose their functionality to form the G4 structures. And when you mutate both, it completely lost their ability to form these G4 structures. Well, further validated by using 2 different methodologies. This is the CD spectra. Similarly, instead of running the gel, we read it in the CD spectra. There's a differences between the positron and lithium condition, indicating that there is a G4 structure forming by these 3' tDRs. Control doesn't have a differences. The completely mutated group has a reverse one, so indicating that we form a G4 structure potentially. This is the direct evidence. We use NMM, which is a G4 structure probe, specifically label all the G4 structures in the gel. So when you stain the gel using the NMM, you will see the bands, positive bands, indicating these are G4 structures. So from the bands, we can tell that these 3' tDR not only form 4 strands G4 structures, but also forming other, like I think there's 2 strands, 3 strands, and even more 6 strands, 8 strands. So what's the function of this forming G4? G4 is a very stable structure. It previously reported it has distinct binding mechanism. It has a very distinct stability. What you can tell from here is very simple assay. We assemble the G4 structure using the tDR mimics, and we incubate with crude cell lysate for 16 hours. Usually, in 16 hours with crude cell lysate, it contains a lot of RNAs will degrade every RNA inside the reaction tube. This is the control mimics. As you can see, there's no leftovers. But when we assemble the assemble the tDR mimics at the G4 conditions, these assembled G4 structure is resistant to the RNAs. This is not true when we assemble using a lithium condition, which is not forming a G4 structure. It was degraded. Similarly, when we mutated, it also lose that ability to form the G4 structure and also the stability. Functionally, the mutated tDR mutants doesn't have the ability to form the G4 structure, lose their ability to inducing autophagy flux as well. So next question is, we already identified the key motifs required for forming this G4 structure. Next, we want to know how. So we calculate the binding proteins of this tDR using a binding conjugated tDR mimics and pull down all the binding proteins, characterizing using proteomics. Interestingly, we identified several 169 binding proteins in the proteomics data. But very interestingly, the top 3 are all PUS genes, which is pseudouridylation synthesis. It's required or is involved in the catalyzation of the pseudouridylation in all RNAs. I think you may have heard that with the COVID vaccination, all the in vitro transcribed RNAs consider this pseudouridylation to increase their stability, decrease their immunoreactions. We first validated using Western blot. Then we designed 2 individual shRNAs targeting all 3 top 4 targets. Try to see which one is responsible or the key downstream partner proteins for the function of this 3' tDR. As you can see here, I didn't show the others, but we only find PS7 shows some interesting phenotype. As you can see here, when we knocking down PS7 at the baseline condition, it's already increased the LC3B lipidation, and it's also dose dependent. This has lower knockdown efficiency, it has lower induction of the autophagic flux. And when further introduction of the 3' tDR seems has compromised the effect of inducing autophagic flux, indicating that, PS7 silencing itself already phenocopied the 3' tDR functionality. We further validate it by using more rigorous strategies by treat the cells with beflomycin. As you can see here, under beflomycin treatment, the lipidated LC3B level was even higher than the scramble tRNA group, indicating that knocking down of PS7 also increased autophagic flux. We also overexpressed PS7. Seemingly, it had the same phenotype as ASOs targeting tDRs. It blocked autophagic flux. So indicating that 3' tDR intact with the PS7, which is required for their functionality. PS7 is usually, as I mentioned, it catalyzing the RNA pseudouridylation. It's involved in different pathways and also involved in different RNA species. The most well-established one is for PS7 specifically, it's small nuclear RNAs, tRNAs, and mRNAs. It has a very conserved motif, UAG motif. So we want to know what's the target for PS7 in this specific case when we transcribed this 3' tDR and the regulation of autophagy. So we did two different profiling. The first one is small RNA profiling. Instead of using normal RNA-seq, we have been using a low bias small RNA profiling that I mentioned in the background, which is called autophagic. We are able to capture all the hypomodified tRNA species, especially small RNAs. So in this case, as you can see here, all the mature tRNAs, small nuclear RNAs, ribosomal RNAs, there's not much downregulation or regulation itself. But in the long RNA profiling, which include both mRNA and long non-coding RNAs, there is a very interesting population is the downregulated groups. They are all histone mRNAs. So histone mRNAs is a very interesting RNA population. It has very dynamic regulation, and the half-life is around 15 minutes to 1 hour, but the protein is very sustainable. So we first validated using qPCR. As you can see, it's all downregulated. And interesting, the protein doesn't change. Some went up as well. When we knock down PS7, it phenocopy the gain of function of 3' tDR. The histone mRNA goes down as well. So as I mentioned, we have a regulation, all of these, either overexpression of this 3' tDR or silencing of PS7, it all regulate autophagic flux. So we are trying to see whether this downregulation of these histone mRNAs has a relationship with autophagy. So we treat the cells with chloroquine to block the degradation of the cargo inside autolysosomes. So as you can see here, when we treat the cells with chloroquine, all of the histone mRNAs, most of them, was rescued. The levels was rescued, indicating that potentially these histone mRNAs are targeted to autophagisome or autophagisome lysosomal pathway for degradation. To further confirm this, we treat the cells with we transfect the cells with tDR immediately, like 6 hours before collecting. We treat the cells with chloroquine to block all the degradation. And then we did a FISH assay. Using the histone probe to identify the histone mRNAs using a lysosomal antibody, LAMP1, to label all the lysosomes. As you can see here, when we transfect the 3' tDR, of course, with the blockage of the lysosomal the degradation, you will see a lot of accumulation of these histone mRNAs in lysosomes. Supporting that, and when we transfect the 3' tDR, these histone mRNAs are targeting to autophagic lysosomal pathway for degradation. So this demonstrate or support this is the RNA autophagy pathway. When go deeper, interestingly, as I mentioned, PS7 has a UGUA-R motif, which is very conserved across all the different RNA species and as a preferred targets. Usually PS7 transfer this U to pseudouridylation in this motif. And you can see here, all of the histone mRNAs has either 1 or to 19 different motifs. And we also checked the pseudouridylation level when we transfect the tDRs. As you can see here, the pseudouridylation, of course, we block the degradation of these histone mRNAs and checking the pseudouridylation level using pseudouridylation IP. As you can see here, transfection of this 3' tDR block or decreased the pseudouridylation level of these histone mRNAs. More specifically, we also further introduce back the PS7 because the hypothesis is the 3' tDR sequester PS7 from these histone mRNAs. So the histone mRNA cannot be pseudourylated. So we transfect back the PS7 protein. This is the white type, the one. The yellow one is PS7 dominant negative with a mutation that cannot categorize the pseudouridylation. So we check both the, this is the H4C3 mRNA. This is the UGUAG motif at the 3' end, I think the 3' UTR area. So as you can see here, only in the UGUAG region, the pseudouridylation levels are regulated by the tDRs and rescued by the PS7 overexpression, but not the mutated PS7. And in the non-UGUAG region, which is the 5', there's not much pseudouridylation detected. So to further confirm, because this we designed a very interesting assay that we transfect the biotin labeled tDRs into the cells. It all binds to the PS7. So we pull down all this complex, the PS7 and the biotin conjugate tDR mimics from the cells, then treat them or incubate this complex with either in vitro transcribed histone mRNAs without any modification, because it could theoretically the PS7 will categorize the binds to the histone mRNAs and characterize the pseudouridylation of these uridine inside the mRNAs, or incubate with tDR cell, try to see the binding affinity between of the PS7 between tDRs and histone mRNAs. So as you can see here, by treating them with different dose of histone mRNAs, it has some competitive binding, means the PS7 could bind to the histone mRNAs for sure. That's its targets. But it is much less than the binding of PS7 and tDRs. As you can see here, when you incubate this complex with tDR mimics, it competitively binds most of the PS7 there. So further, because we demonstrate that the PS7 cannot pseudourydylize the histone mRNAs, so the pseudouridylation deficient histone mRNAs are targeted into lysosome for degradation. To further confirm this, we leverage the in vitro transcription by incorporating either pseudouridylation or fluorouridylation, which cannot be pseudourylated. So we transfect these, we in vitro transcribe the histone mRNA with 2 different modifications. Like this one was recently used for all the mRNA vaccines, but this one cannot be pseudourylated somehow. When we transfect the fluorouridylation modified histone mRNAs into the cells, it induces the autophagy. or increase autophagy flux, I would say. So that means the pseudouridylation deficient mRNAs, especially the histone mRNAs, can induce RNA autophagy. This is also true in vivo. I didn't show that in the mouse models, we have a very nicely negative association between the tDR levels with histone mRNA levels. This is the direct evidence that when we transfect the tDR mimics or deliver the tDR mimics into the kidney, as you can see, most of the histone mRNAs are downregulated. In the human, we also validate in the human tissues as well. This is a collaboration with Marco Spicer from the University of Michigan. He has a large cohort that collect the kidney tissues from different kidney diseases. We separate them into two different populations with the EGFR larger than 90 considered as kind of healthy, and the EGFR less than 60 will claim that as chronic kidney disease, especially in the earlier phase. So as you can see here, the tDR levels is higher in the kidney disease tissues. It's also true that the histone mRNA is also decreased in these kidney tissues as well, indicating a negative association between the tDR and these two histone mRNAs in human tissues. We also have some preeclampsia patient data. I didn't show it here, but it also shows a negative correlation. So in summary, I want to calculate maybe easier for you to understand. In the baseline condition, healthy condition, the tRNA was mostly involved in the translation. And all the histone mRNA, nascent one will be pseudo-related and target into ribosomes for translation with the involvement of the tRNA for sure. But in the metabolic stress condition, these tRNAs getting cleaved into tRNA fragments, we call tDRs. And these tDRs, in this specific case, tRNA species of 3' tDR can form a very stable intermolecular G4 structures. And this G4 structure are able to sequester PUS7 from the original targets, which is histone mRNA targets. So these histone mRNA targets cannot be pseudorylated. The pseudouridylation deficient histone mRNAs then target into lysosome for degradation, triggering RNA autophagy pathway. So on to summary here as well, we have identified that tRNA tDRs is a novel class of small regulator RNAs. Studying tDR is very challenging, but I believe with the development of several advanced tools, it will be more feasible in other groups as well. In this specific case, we have identified this specific tDR induced by hypoxia can activate autophagy flux in kidney cells specifically. And this 3' tDR activates RNA autophagy by sequestering PUS7 protein from its target histone mRNAs. These histone mRNAs cannot be pseudorylated, and then they will target into autophagy pathway for degradation. The administration of the 3' tDR, most importantly, can protect the kidney from injury. So this is the key we want to explore further is, the chronic kidney disease, what we can saw from the tissues, either from the human data, we analyze the tissue from earlier phase. But in the mouse models, when we check the later phase, when it has developed chronic kidney disease, this 3' tDR levels was downregulated, or even compared to is much lower than the control groups. So we will working on deliver this tDR using different formulations and different chemical modifications to try to restore the level of this 3' tDR in the kidney tissues, try to restore their autophagy activity, and then we'll try to investigate whether these restoration of 3' tDR and autophagy can protect the kidney against the chronic kidney injury. So I want to stop here and thank my lab at Mass General and also my collaborator, previous mentor, Soumya Das, and the collaborators from Brigham, UCSC, and University of Michigan, and the funding agency from American Heart and NIH. I'll stop here and happy to take any questions if you have.